So let's get started with the final session. And I'm hoping this is going to be the crescendo of the day, where we bring together many of the concepts that we've been discussing throughout the day. And the way we're going to organize this is in three parts. First of all, we're going to invite a speaker to present an example of a potential disruptive technology or change that could enable drug discovery, and use that exemplar to stimulate discussion across panel members about that question that I asked earlier. What would be a disruptive change to developing medicines from genetic insights? So first of all, I'll invite Dan to kick us off. With a kind of a special seminar, we were given him a pretty difficult task in 10 minutes. Summarize and exemplify a disruptive technological change that could really enable drug discovery. And we've asked Dan to talk about a specific technology, really to set us up that other people can then react to. So I'll hand over to Dan.
All right, thank you, Nadine, and certainly thank you to the organizers, in particular Jeff Ginsberg, for the opportunity to talk about tissue chip technology. So tissue chip is essentially a program that is funded in part by the Cures Acceleration Network, which is a program that was put in NCATS, National Center for Advancing Translational Sciences, when NCATS came into being in 2011. And Jeff and Frida Lewis Hall are the co-chairs of the review board that oversees this program at NIH. And so what I'd like to do is to give you an overview of this program and how we're making this to be a paradigm shift in how drug discovery is made, not only in terms of evaluating safety, toxicity and efficacy of candidate therapeutics, but also in some ways how regulatory agencies can make really true decisions in terms of effectiveness of drugs. And so as Nadine said, this is a difficult challenge because it's a very complicated program. And so what I'll do is just to give you a brief overview and then really show a video which we have developed to hopefully bring home the message. So essentially the central goal of this program is to develop an in vitro platform that is heavily using human tissues to evaluate safety, efficacy, and toxicity of any promising candidate drugs. So the goal is to capture the 10 major organ systems of the body, as listed here on this slide, and then recreate them in microfabricated, essentially engineered devices, microfluidic devices in most cases, and have them be physiologically relevant, also be genetically diverse, and of course pathologically meaningful. We want this to be modular, and you'll show that, you'll appreciate that more in the video, meaning they're kind of like Lego blocks that you can plug and play. And because of the need to do repeated testing for toxicity, we require that the tissues actually survive for at least 4 weeks. We have platforms where some of the tissues are actually surviving for almost 3 months. And of course, at the end of the day, we want this to be widely available to the disease community. So here's a brief video that was developed to help explain the program a little bit more.
Several thousand diseases affect humans, of which only about 500 have any treatment. Getting a drug through the current development pipeline takes too long, is too costly, and the vast majority of new therapies, about 95%, never make it past clinical trials. Drugs that seemed promising in early studies using cell and animal models often don't have the same response in humans. We need an alternate research model that accurately represents the human response. NCATS, the National Center for Advancing Translational Sciences at the National Institutes of Health, is supporting research to make this possible. Through the Tissue Chip for Drug Screening Program, a collaboration among NIH, DARPA, FDA, and researchers nationwide, NCATS is supporting research on bioengineered devices that mimic the function of human organs. The goal is to use these tissue chips to test the safety and effectiveness of potential new drugs, weed out those that don't work, and pursue ones with the most promise. The chips are fabricated from a flexible plastic with ports and channels to allow nutrients and oxygen to pass through them. Unlike cell culture and animal models, the innovative design of tissue chips replicates the complexity of the human body environment. One example is a liver chip, which includes multiple cell types that are organized much in the same way as they are in the human liver. Features such as blood flow can be mimicked to recreate what cells experience in the body. The complexity of these chips generates human-specific data, which enables researchers to better understand how the liver responds to a drug and if it is toxic, critical data before testing in people. By connecting chips that represent various organs into a full human body system, scientists may better understand how the whole body responds to drugs and if they're safe and effective. For example, chips that include additional factors such as hormones could help researchers investigate differences between male and female responses to drugs. The ultimate goal is to use these chips in a variety of ways, including to advance precision medicine, test for environmental toxins, and develop disease-specific models such as for cancer and rare diseases. By developing, demonstrating, and disseminating tissue chip technology, NCATS is improving health through smarter science.
So tissue chips is really a product of convergent science. So it takes a team of engineers, material scientists, in this case cell biologists, and we essentially use a lot of iPS derived stem cells for our seeding the fabricated structures. And then, of course, we try to recreate the gradients of hormone and cytokine response. And an important part, which I'll show an example for, is perfusion. Because as you imagine, as we're trying to recreate the 3D cytoarchitecture of tissues, these are not just your regular 2D culture or even an organoid culture. So you need microvasculature. You would need dedicated bioreactors that would have essentially be optimized for the right tissue response, innervation, and I'll show you an example of that as well as the immune response. And of course, we are looking at non-invasive, real-time readouts that is not necessarily based on biochemical or cytopathological analysis. And then, of course, being able to improve on the system by evolving the technology as time goes on. So one of the examples I'll show is the microvasculature. This is work by Stephen George. And this is derived from IPS line. And you can see here 1 micron beads flowing from the bottom to the top, simulating a vasculature bed. And then in the middle, you can actually, oops, went too fast. You can then embed tissues, whether it be cardiomyocytes or other, in this particular instance, is a colon tumor that was being perfused in this particular vascular bed. And so you can study the effect of chemo drugs in terms of angiogenesis or anti-angiogenesis effects. This is what the vascular tissue chip look like. So you can see that there are a number of vascular beds, and you can embed several different tissues in there in a series, being able to look at organ crosstalk in some ways. The other example that I'll show would be innervation. So in this case, looking at the work of James Wells from Cincinnati. So I won't go too much in terms of reconstructing the GI system on a chip, but when we have the GI system on a chip co-cultured with vagal nerve-derived neural crest cells, what you end up forming are the mesenteric and myenteric nervous system and mucosal nerves, and this would be something that is important in terms of simulating peristalsis. So this is the gut on a chip undergoing peristalsis as part of the innervation on this system. So these are really quite sophisticated 3D cultures on chips. And we can also show that this also secrete the digestive enzymes as shown here. Crypts and villi are formed using EM microscopy, and also being able to culture the microbiome limited to into the gut in these systems. Another example that I show is the heart on a chip, in this case using a cantilever device to simulate the contractility of the heart or the cardiomyocytes. And this was actually used by Kit Parker's group from Harvard to model a rare disease called Barth syndrome, which is a disease causing cardiomyopathy due to a deficiency in the mitochondrial membrane. So this is an example of the cardiomyocytes from an iPS-derived healthy individual, and this is from a Barth syndrome patient. This was from a Barth syndrome patient, iPS line rescued by cDNA transfer, and the platform was actually used to understand that Barth syndrome is now caused by ROS accumulation, and is now being used to screen for drugs of ROS scavengers as a potential treatment for Barth syndrome. Another example is on progeria, Hutchinson-Gilford progeria syndrome, which is a premature aging disease, primarily caused by loss of lamin A. But you can see here, it leads to an abnormally shaped nuclei, as shown here. And so our investigator, George Trusky from Duke University and Can Kao, in collaboration at the University of Maryland, developed this tissue-engineered blood vessel because atherosclerosis is the primary cause of death in progeria. And I'll show you a couple of slides that shows that the fibrotic lesion in the smooth muscle cells is also being recreated in terms of the accumulation or actually calcification in this individual, and then being able to rescue the phenotype with rapamycin. So this is showing that looking at mesenchymal-derived stem cells versus iPS-derived stem cells, you actually get better characterization in terms of apoptosis, alizarin red staining for calcification, and also for alpha smooth muscle actin shown here. So in the case of iPS-derived cells from patients, they have larger diameters compared to normal. This is from mesenchymal stem cells. And then if you treat them with phenylephrine, the iPS-derived lineage show vasoconstriction, and then in response to acetylcholine, it also has a better vasodilation compared to the controls. And then also using rapamycin treatment, in this case RAD001, you can see that with no treatment and with 100 nanometers, you get better representation of smooth muscle, and also calcium binding protein as shown by staining for calponin. This is my last slide, and this is really what I would like to drive home in terms of the potential for tissue chips, including the ability to incorporate a lot of genetic resources that we've been talking about all day today, and being able to capitalize on iPS technology. And we're actually well into the process. The program started in 2012. We're about 1 year left on the first phase of the program to develop the human on a chip, or homunculus, which could certainly be used for a number of different things. But we are also in this year launching a program in terms of creating rare disease modeling on chips, as well as being able to expand that into additional disease models in 2017. And we've also partnered with the Center for Applied Sciences in Space, which is associated with NASA, to develop tissue chips in space, essentially to look at the effect of microgravity and how that could inform us in terms of drug development, especially looking at epigenetic changes. As you know, there's accelerated aging in space, but it's reversible once you go back on Earth. And so those are the things that we are looking for in terms of novel drug targets. And then looking at the druggable genome, essentially through gene editing technology, by looking at IPS lines on an isogenic background and being able to introduce various polymorphisms and being able to dissect out various risk factors. And we have also a plan to expand this to precision medicine by developing the U-on-a-chip system, which is essentially looking at individual drug response. So essentially your own fibroblasts can be made into IPS lines and be able to make your own individual chip system. And then my goal eventually would be to be able to conduct clinical trials on chips, which could then essentially we're trying to contract and become cost efficient in terms of preclinical as well as the clinical space as well. So with that, I'll end, and I apologize if I went too long.